All right, so of course we've got a warm up. It is Unit 3, Day 3. Today is February 4th. So you've got 6 minutes to go ahead and knock it out. All right, so hopefully that was enough time to get the warm-up done. Quick 6 minutes. We emphasize those warm-ups because they give you insight into tests and the questions you will see on these tests. So please make sure you are taking them seriously. I know in the grand scheme of things, you're like, it's just 2 points. What does it really matter? But those are probably the most important 2 points you'll get throughout each unit. So every warm-up, every exit ticket matters. They add up to be just about as much as one of the asynchronous assignments, depending on how long the unit is. But they really, really do give you guys good practice for the exams that we'll take. So in addition to doing them once, perhaps on the day before the test or the couple of days before the test, you'll go back and you'll redo a few of them just to continue to get practice with those questions. Some of them might be very, very similar, if not exactly the same, as to the questions that you will see on those exams because they come from the same source. Keep that in mind. Today's lesson is about meiosis. Yesterday we talked about mitosis, so similar process, important differences. That being said, I want you all, as we move through this lesson, to start to envision a Venn diagram, perhaps even consider drawing one and writing in some facts about each of these processes, some of their similarities and differences. Today is February 4th, 2021, and it is the third day of our third unit. Today's objective is to explain the role of meiosis in sexual reproduction and genetic variation. We'll begin the process in this conversation of talking about genetic variation, but we'll really continue it and dive a little bit deeper into it tomorrow. The essential question that we seek to answer by the end of today's lesson: how does meiosis ensure genetic diversity in sexually reproducing organisms? That should say day 3. All right, so we're going to start off today with a quiz. So you can go to joinmyquiz.com and enter the game code 564802. 564802. Or you can click on this link that I'm about to send directly to the chat. And you'll have access to the quiz that way. Thanks for joining us, Elena. Please go ahead and get ready to join this quiz with us. Be active and present in today's class, please. All right, thank you, Araceli. Thank you, Sally and Janelle and JT and Melissa. Folks are rolling in now. Thanks, Abigail. Thank you, Lisa. Thanks, Josue. We've got 8 people, and there are 15 of us in today's meeting. Okay, let's take about 30 seconds to join. Of course, if you aren't able to join before we start, you still can join as the game gets underway. Okay, we've got 12 folks. So 3 of us are not yet in the game. Continue to get ready to join if you have not yet already, but we are going to start. Okay, folks, I want to see the accuracy stay right where it is. Let's not dip below 70%. I'd love to see it maybe even push up towards 80%. So we're in the right direction. We're trending positively. We're at 74% right now. Let's keep going up. JT's in an early lead, followed by Abigail, Dejan. All of them are on a 4-question streak. Let's stick with it. Let's stick with that. Good job, Abigail. Keep going. Good job, Sally and Janelle. Let's keep that accuracy where it is. Good, guys. I like to see that accuracy where it is. Okay, so I know we're finishing up. We've got 5 people who are still working. Let's take about 3 more minutes to get finished totally, and then we'll talk about some of the questions that we struggled on. But I like the accuracy where it is, 73%. That's excellent. Let's keep it up there. Keep it up. And let's see if anybody can catch Janelle, who's in the lead. I know we still have some folks working, so maybe you guys will be able to catch her. And Abigail, basically almost tied. Okay, so it looks like the top 5 are pretty solidly locked in, but we still have 4 people working. 1 more minute, 1 more minute. Okay, so let's take a look at it. We've got 3 more people working. That's okay. Keep going. So we did really well. No questions are marked red. That's always a good sign. So, okay, so 7 of us got number 2 incorrect. It says, in what phase of mitosis does DNA condense into visible chromosomes? Does anybody remember when that happens? When does DNA condense from chromatin into chromosomes? Okay, so Michelle, you're saying telophase. Um, let me see. Hopefully that wasn't the right answer here. Let me make sure. Oh, I can't click on it because.
I meant prophase.
Okay, prophase. Good. Yeah. Now I can see why someone might say telophase because the opposite actually happens. So let me see if I can find a good picture representing this. All right. Actually, this might be better. So let's take a look at this really quickly. So in this image, we can see that during interphase, the DNA inside of the nucleus is very tangled, it's unorganized, it's chaotic. We call that form of DNA chromatin. During prophase, though, we can see that it becomes organized into specific chromosomes, which are much less chaotic, and it's condensed. So when we go from chromatin to chromosomes, that's prophase. But then at the end, in telophase, those chromosomes kind of fall out of order back into the form of chromatin. So don't be confused about that. To go from chromatin to chromosomes, that happens during prophase. We've got to organize the DNA in order to be able to pull it apart. But then once it's been pulled apart and we've formed two new daughter cells, the DNA will return into its chromatin state. So I can see why we would say telophase, but prophase is when it goes in the correct order that's referred to here. We did pretty well on this one and that one. Let's look at number 5. What are the phases of mitosis in order? All right, so this one was really just a test of, could you remember what we talked about yesterday? Or do you have your notes in front of you? But can someone just tell us what the correct order is? What's the correct order of mitosis? Good, thank you, Anna. Thank you, Melissa. So PMAT, prophase, metaphase, anaphase, telophase, PMAT. You can come up with your own mnemonic devices if you need to remember that. We struggled with number 8 a little bit. More people got number 8 incorrect than correct. At what stage during mitosis does the nuclear envelope break up? Does anybody remember that? When does the nuclear envelope break up? Good, thank you, Melissa. Prophase. So this is, again, one of those earliest processes. Before we can actually have access to the DNA, we've got to make sure that the nuclear envelope has been broken apart. It needs to dissolve. Otherwise, the DNA is going to be protected by that nuclear membrane. So before the spindle fibers can actually reach the chromosomes, the nuclear envelope has to dissolve. It has to disappear. And that happens during prophase, the very first step of cell division. All right, any others that we struggled with? Let's see. All right, we did pretty well on all the others. So good job on that. I definitely love where our accuracy is right now. It's at a 73. I know we still have, looks like one more person working. So that person can finish up and we'll get some even better statistics from it. But good job, everyone. That's where we want to be. Mitosis is a foundational topic and we're going to build on it today, talking about meiosis. But that was a strong start. Okay, so we had the same practice problem yesterday, and I asked you to place these images in their correct order as they happen in cell division. This time, I don't want to worry so much about the order, but I want you to tell me what the name of each image is. So in this first image, what step is happening here? What's the name of this step? Good, thank you, Abigail. And how do you know that it's anaphase? How can we, yes, good. So ana, the prefix in anaphase, means to literally pull apart. We can see that the sister chromatids here are being pulled apart and the cell is starting to stretch, right? Because we're starting the process of forming two distinct separate cells. Good. What about number 2? What does number 2 represent? Yeah, prophase, excellent. No, I'm sorry. There's another one. JT, you're close to the right answer, but there's a difference between this one and prophase. So that one even that tripped me up. So I apologize. But what is what happens before prophase? Yes, thank you, Abigail, for cleaning that up for us. This is interphase. And we're going to discuss why this is not prophase and the difference between this picture and the prophase picture shortly. What about number 3? This is probably the easiest one to identify. Thank you, Anna. Yeah. How can you tell, Anna, that this is metaphase and JT? How can you tell? Okay, we see the chromosomes, and what's special about the chromosomes in this picture? They're starting to line up in the middle of the cell. The prefix meta literally means middle. So when we see these X's, these chromosomes lining up in the middle of the cell, that is a depiction of metaphase. What about number 4? This one's prophase. Good. Now, JT, I made the mistake of saying that the number 2 was prophase initially. There's an important difference that we should note. So as we just discussed in that quiz, during prophase, the nuclear envelope is going to start to dissolve. So in picture number 4, we can see that the nucleus is represented by a dashed line. Typically, that's how we represent a nuclear membrane that is not totally there. It's either dissolving or it's being formed. So we know that this is prophase because we see that this nuclear membrane is starting to disappear. We can also see that the centrioles and the spindle fibers, which remember, that's like the fishing rod that's going to pull in and separate the chromatids. We can see that they're starting to move into place. So that's another indication that this is prophase. And then lastly, number 5, which step must this be? Telophase. Thank you, Anna. Yeah, we can tell because, again, we see that the nuclear membrane is a dotted line. It's a dashed line. So the nuclear membrane in this case is actually reforming, but we can also tell because the cells are separating. They're being pinched together in the middle of the cell, and eventually they will move away from one another in cytokinesis. Okay, so I love these images. In all of my science classes that have been image heavy, I've found that it benefits me to actually reproduce the pictures, to draw them on my own. And if we were in person, then that would be one of the things that we would do. I'd give you guys scratch paper and colored pencils and whatnot, and you would draw out these different steps of mitosis. But even though I can't do that in person right now, you all should still take time to try to draw these different steps and phases out. Give yourselves a mental representation of what is happening. I promise you that will help you to remember better what happens in each of these steps. So I'll leave this up if you want to take a screenshot or a picture over the next 10 minutes or so. Not 10 minutes, I meant 10 seconds. My brain is a little slow. I'm kind of tired today. Okay. So when we're looking at this image, this is not some alien planet or some futuristic device. What is actually happening here? What's happening in this picture? Good. Thank you, Abigail. And this is indeed the process of fertilization. How can you tell? What are we seeing in this picture?
Sperm cells all around a egg.
Good. Thank you, Lisa. Yeah, we're seeing sperm cells that are kind of swirling around this larger egg cell in the middle. Question that's going to be a throwback question to our previous unit, unit 2. What are these long tails called that the sperm cells have? Excellent, JT. Yeah, those are called flagella. Good. So I just wanted to test and see if you guys could still remember that unicellular adaptation. But we see these sperm cells. We're going to talk a lot more about fertilization in an upcoming unit. Today, we're going to talk a little bit about the process that allows fertilization to happen, the process that produces sperm and egg cells. Before we do that, I just want to talk a little bit more about this image. What's happening is that all these different sperm cells are trying to essentially embed into this egg cell. But probably what's happening is one of them was already successful in doing so. The process of fertilization is really quite remarkable for really small details. One of the details is that the egg cell will actually close itself off to all the other sperm cells once one of them has successfully penetrated the egg cell. So it creates this protein coating around its outside the moment that one sperm cell is able to get in. And from that moment forward, what you've got is this process of development. That sperm and that egg are forever linked. They share DNA now. And over time, they will become a zygote, then they'll become an embryo, then they'll become a fetus, and then a human child, or whatever. We can't assume that this is human sperm and egg, but whatever animal this is, it will start to develop into that animal. So we'll talk more about that, but I think it's really cool. You know, the really small details that make fertilization possible are quite remarkable. Okay, so the process that allows fertilization to happen is called meiosis. Meiosis is the process that the process that produces sperm cells and egg cells. And remember, we call sperm cells and egg cells gametes. What are all the other types of cells called? We talked about this yesterday, but if a cell is not a gamete, then what must it be? What type of cell must it be? Good, thank you, Abigail. So gametes are our sex cells, and thank you, Janelle, you jumped in too. Everything else is called a somatic cell. Soma, S-O-M-A, is a prefix that means body. So all of your body's other cells, we've got hundreds of types of cells in our body. They are somatic cells, and they're special because they have your entire genome. They have all 46 chromosomes. These gametes, on the other hand, are special. Your sperm cells or your egg cells, they're special because they have half that number. Instead of 46 chromosomes, they only have 23. And we're going to talk about that as well. But it's important to note two things. Number one, interphase still has to happen before meiosis. Just like interphase comes before mitosis, interphase has to come first. We talked about the reason why. We've got to double the DNA. We've got to make a copy of the DNA. The other thing that's important to note is that meiosis itself is not sexual reproduction. Meiosis is used for sexual reproduction. It's used to create sperm and create egg cells, which then can be engaged in sexual reproduction. But meiosis itself is not sexual reproduction. It just produces the things that are necessary for sexual reproduction. All right. So again, as I said at the beginning of the lesson, I want you all to start to create this Venn diagram. You can literally write a Venn diagram, two circles that overlap in the middle in your notes, and start to compare mitosis in one circle, meiosis in the other. They've got crucial similarities, but they also have crucial differences. We talked about mitosis yesterday. Mitosis was one round of cell division. Meiosis is 2 rounds of cell division. And I'll talk about exactly what that means in a coming slide. Mitosis was used to make 2 daughter cells. Meiosis is used to make 4 daughter cells. Mitosis was used to make identical daughter cells. Meiosis is used to make unique daughter cells. And then one more difference, mitosis was used to make diploid daughter cells, and meiosis is used for haploid daughter cells. So each of these words that you see here is basically different than what we talked about for mitosis. Thanks for joining us, Sabina. Okay, and a haploid cell is a cell that only has half the total number of DNA from a somatic cell. So whereas the human somatic cells all have 46 chromosomes, human haploid cells only have 23. All right. So how do they get to this point of only having half the number of chromosomes? Well, what's not shown here is that before we get to 46, we still need to go through a process of interphase, which replicates our DNA. But we're going to start off with 46. Again, meiosis has 2 rounds of cell division. So after the first round, we get down to 23. And then it's going to undergo another round of cell division to produce 4 totally unique cells. So each of these is genetically unique. This is why 2 people can come together and have 20 children, and each of their children will be different. None of their children will be exact clones. You could have 1,000 children if that were physically possible, and none of them would be exactly the same. They would all have different physical and even different psychological and mental and emotional characteristics, because those things are all influenced by our genes. We're going to talk about some of the processes that lend themselves towards genetic variation tomorrow. So genetic variation is guaranteed because of this process of meiosis. And we'll talk about exactly what happens to get to that genetic variation. So now we've got really 2 different terms that we can use to refer to sperm and egg cells. We can call them gametes. I guess we got 3 terms. We can call them sex cells, and we can call them haploids because they have half the number of chromosomes, half the amount of DNA as the parent cells. So sperm and egg cells could be gametes, sex cells, or haploid cells. So again, 2 rounds of cell division. So we've got meiosis 1, which has prophase 1, metaphase 1, anaphase 1, telophase 1, And then there's some cytokinesis. We split into two cells, but it undergoes that process again in meiosis 2. So prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis again to eventually give us 4 daughter cells. All right, let's take a look at this image. So we start off with a cell, single cell, a parent cell. This parent cell is a diploid. It's 2N. 2N represents diploids. During interphase, its DNA gets copied. And so what happens is that we end up with twice the amount of DNA. So in humans, we start off with 46 chromosomes. We copy it. Now we have 92 chromosomes. We doubled that amount. What we also do is we set ourselves up to have what are called homologous chromosomes, chromosomes that are paired up. So, for example, you get a chromosome number 1 from your dad and chromosome number 1 from your mom. You pair up those 2 chromosomes. You get a chromosome number 5 from your dad and a chromosome number 5 from your mom. Those get paired up. Chromosome number 13 and chromosome number 13, they get paired up. So all these chromosomes that have the same genes on them, that's how we decide who gets paired up. They have the same genes. So the gene that decides for hair color is located on a specific chromosome. Those chromosomes will be paired up next to one another. During anaphase, the homologous chromosomes get pulled apart, and then they get separated into different cells, into 2 different daughter cells. That happens during anaphase 1. During anaphase 2, because now we only have, one of those homologous chromosomes in each cell, it's now the sister chromatids that get pulled apart. So instead of pulling apart the chromosomes, we're actually basically cutting the chromosome in half, and we're pulling apart the sister chromatids. So that's an important thing to note, an important difference. Anaphase 1, chromosomes are pulled apart. Anaphase 2, sister chromatids are pulled apart. Okay. One other thing that we'll talk about, and again, tomorrow is a discussion about genetic diversity, genetic variation. There's a process called recombination. More commonly, we refer to it as crossing over. That happens in prophase 1. So when we line up those homologous chromosomes, you get chromosome number 8 from your dad, chromosome number 8 from your mom. We line them up, we pair them together. They exchange some DNA. So a little bit of that DNA from your dad is going to switch over to the chromosome from your mom. A little bit of your mom's DNA is going to switch over to the chromosome from your dad. And what this happens is what this creates is that each of the chromosomes is now just slightly different from one another. Different combinations of your mom's and your dad's DNA on different chromosomes. That results in eventually 4 daughter cells that are totally unique. They have different characteristics because they've gotten different combinations of genes from each parent. Keep in mind, in this picture, we're only seeing 1 chromosome. This is only representing 1 chromosome, but humans have 23 chromosome pairs. So the different possibilities are humongous. This is why it's impossible for, even if a person had thousands of children with the same person, all of those children would be slightly different from one another, because the possibilities are endless with this number. Not endless, you could actually calculate it, but it would be a number in the billions in terms of how many different combinations you can end up with. All right, so again, we did this yesterday, but we're thinking about some of the math involved here. This top cell would be referred to as a germ cell, literally like germs, but it has a different meaning. GERM. Germ cells are the cells that actually produce, they will eventually become sperm and egg cells. They are the cells that go through this process of meiosis to produce sperm and egg cells. There's an important difference between men and women, or I shouldn't say that, between males and females. Male animals will be producing sperm for their entire lives. Now, when you're younger and you're more fertile, male animals produce more sperm. They're more efficient with it. But even until they are well into, in the case of male humans, well into their 80s and 90s, they are still producing sperm. Female animals are born with a finite number of eggs. That number varies between different animal species. It can even vary within the human species. It can vary between women of different ethnic backgrounds based on their diet, essentially. But a female animal is born with a set number of eggs, and that's the total amount of eggs that she will have throughout her entire life. So from the time that she first begins menstruating until the time when she's done menstruating, a period that typically lasts between 25 and 40 years, she only has a set number of eggs. So it's different processes, but they each come from what's called a germ cell. And that's what would be represented here. That germ cell is the original parent cell, and it undergoes meiosis to eventually produce these gametes, which are genetically unique. But what happens, we go through interphase, just like we talked about yesterday with mitosis, that replicates the DNA, so we get twice the number of chromosomes. Then we go through meiosis 1, we end up getting back down to the diploid number of chromosomes, and then we undergo meiosis 2, and we end up with haploids, 4 haploids that have half the number of the original parent cell. So in a koala, there are 16 chromosomes. So after interphase, how many chromosomes will that cell have? After interphase. Good, thank you, Abigail, 32. And then what about after meiosis 1? Yeah, good, thank you. So after the first round of cell division, after meiosis 1, we end up back down to 16. And then after meiosis 2, how many chromosomes will there be? Thank you, Josue, 8. That's why it's important that we go through 2 rounds of cell division. The reason why we want to end up with half the number, the reason why it's important that we have haploids is because the egg cell and the sperm cell combine. So if they combined and they doubled the amount, then there would be too much DNA. So they need to start off with half the amount to end up with the right number. Now, one of my students in my regular class brought this up yesterday, and it was a really, really good question. So I want to mention it to you all. But she said, what if you have the wrong number, essentially? So what if one of them ends up with, in this case of a koala, maybe 7, and the other one ends up with 9? That is called a mistake, and that does happen. And that can lead to sometimes fatal disorders, sometimes disorders like Down syndrome is a result of that happening. In that case, usually it's the egg cell that ends up with a little bit too much DNA, 1 extra chromosome. And so instead of having 2 chromosomes in the 21st position, that person who has Down syndrome has 3 chromosomes, and it's called trisomy 21. It means they have 3 chromosomes in their 21st position. It could also happen in the 23rd position, trisomy 23, in which a person ends up with 3 X chromosomes. Instead of 1 X and 1 Y, or 1 X and 1 X, they end up with 3 X's. And that can lead to a disorder called Klinefelter syndrome. And typically these people are female, and they are a little bit taller, and they might be more subject to some mental disorders. But it is possible for there to eventually, in the case of something that has gone wrong, for there to be more or less chromosomes in one of these gametes than there should be. Now, it's important that we note, like a koala, for example, a koala cannot mate with a kitten or with a cat because not only do they, obviously there are some other reproductive barriers. They don't have the actual machinery that would go together, nor do they have the desire. But genetically, it also doesn't work because let's say that this koala bear has its gametes have 8 chromosomes. A cat, I don't know the exact number, but I'm just going to make it up. A cat might have 20 chromosomes in their gametes. So that number is too far off for it to work. You all will have a practice problem. I can't remember if it's in today's exit ticket or tomorrow's, but there is a practice problem that will kind of show you exactly what I'm talking about. Different species have different numbers of chromosomes. And so that's one of the genetic barriers that prevents them from mating. That's why, for example, if you cross or if you take a horse and a donkey, when those two things mate, does anybody know what comes out of it? The horse and the donkey.
A mule?
A mule. Good, Lisa. Yeah. So if you cross a horse and a donkey, you get a mule. But that mule we can't consider mules their own species because they cannot reproduce. Mules are totally sterile, and it's because of the genetic mix-up. Horses have a little bit, I think, a little bit less, a little bit fewer chromosomes than donkeys do. So even though those two things can mate, the result is that mules do not have the genetic capabilities of having children themselves. So it's an interesting process, and you'll have a practice question that will show you exactly what I'm talking about. All right, we've got 16 minutes. The exit ticket is, of course, 4 questions, multiple choice. The asynchronous assignment today, U3D3, Meiosis practice, is also multiple choice questions. I just made it, but I think it's only 12 or 13 questions. So you all should be able to get both of them done today in the next 15 minutes. And then if you weren't able, for whatever reason, to do one of the previous asynchronous assignments, I definitely encourage you to do that. All of our assignments will be due in 6 days on February 10th, next Wednesday, and that will be test day. So just keep that in mind. I will be here on mute if you have any questions, but go ahead and use this time wisely to go ahead and get your work done so you don't fall behind. Okay, folks, it's 1:20. So I thank all of you who are working diligently just now during the asynchronous portion of class. Go ahead and enjoy this next 10 minutes before 4th block. We've got one more day this week. So let's take some positive momentum into it and accomplish what we need to accomplish one more day, and then we can relax this weekend. Talk to you all tomorrow.